Everyone. Now that we know what a skeletal muscle cell looks like and what a sarcomere looks like, the next two videos focus on the actions that happen cellularly for the skeletal muscles to contract and to relax. So in this part one, we will focus on what happens at the neuromuscular junction and the events that lead to an end plate potential. And the other key learning objective here is to describe what excitation-contraction coupling is. So let's get started. So here's a cartoon that should look familiar. So we're looking at a sarcomere. So remember that a sarcomere goes from a Z disc right here to another Z disc right here. And as we're accustomed to, we see the actin in red on the outside of the sarcomeres and the myosin in purple in the middle of the sarcomere. But what I really want to bring your attention to is the size of the sarcomeres. So sarcomeres can lengthen and shorten. So let's compare the size of the sarcomere at the top here, which goes from here to here, to the size of the sarcomere at the bottom. And you can clearly see that the top sarcomere is longer than the bottom sarcomere. And this is what happens with skeletal muscle contraction. So the sarcomere at the top would represent the position of a sarcomere in a relaxed muscle. When the muscle isn't contracting, the sarcomere is relaxed and it's longer. You can see there is some overlap of myosin with actin, but overall the overlap is pretty small. That's in contrast with a sarcomere in a contracted muscle, which is much smaller. And you can see that the overlap between myosin and actin here is much more important. So how do we get from a relaxed sarcomere to contracted sarcomere? It's through the action of cross bridges. Remember that cross bridges is the interaction of myosin with actin. So when myosin and actin can physically interact with one another, myosin actually pulls the actin towards the center of the sarcomere. So you can think about it as the action of a tug of war, where myosin pulls on the actin little by little and brings the actin closer to the center of the sarcomere. So that's how we go from a sarcomere that's this long to a sarcomere that's much shorter. But the real question of this video is, how does this happen inside the cell? And through the next two videos, we'll talk about three major steps. The first one is that acetylcholine must depolarize the muscle cell. And it does so at a very specific location, which is the neuromuscular junction. So neuromuscular junction is a fancy word to say a synapse between a neuron and a muscle cell. So we're quite familiar with synapses already. So think of a neuromuscular junction as the specialized synapse, but instead of being between two neurons, it's between a neuron and a muscle cell. So let's dig a little deeper into this concept. So this is a review of the nervous system and how it's organized. We've already discussed previously in the course that within the peripheral nervous system, there's two types of neurons. There are autonomic neurons and somatic neurons. Remember that the autonomic neurons are not under voluntary control, and they regulate the actions of glands, smooth muscles, and heart, for instance. That's in contrast with our somatic neurons, which is what we'll see in this video. The somatic neurons only have one target, and they are skeletal muscles. So all of the skeletal muscles that we have are controlled by somatic motor neurons. So let's focus on the cartoon here, especially here. So this is a cartoon of a neuromuscular junction. Remember that specialized synapse between a neuron here, specifically a somatic motor neuron, and the muscle cell. And we're describing what happens at this junction. So all somatic motor neurons uses only one neurotransmitter, which is acetylcholine, or ACH. So when this motor neuron is activated, it will release acetylcholine at the neuromuscular junction. Now, the specific location where that's released on the muscle cell is called the motor end plate. And we'll look at this a little better in a few slides. As we know, acetylcholine is an excitatory neurotransmitter, meaning that when acetylcholine binds to its receptor at the motor end plate, the result is to depolarize, right? Depolarize or activate the muscle cell. This depolarization is not yet an action potential. The first depolarization that happens at the motor end plate is called an end plate potential. And these end plate potentials are graded potentials. And they come before action potentials. So the end plate potentials are the first depolarization. The large majority of them will reach threshold, and that will initiate action potentials that will then depolarize the entire muscle cell. So this is a slide that we've already seen in the neural section of the course. Let's remember the nicotinic synapse, or the synapse that uses acetylcholine. Because what happens in this neuron, in the presynaptic neuron, is the same thing that happens in the somatic motor neuron at the neuromuscular junction. So remember, when we discussed the synapse, we had an action potential that would depolarize the axon terminal. Then voltage-gated calcium channels located in the presynaptic neuron open, and that triggered the influx of calcium. And that was needed in order to signal for exocytosis of the vesicles containing acetylcholine. When acetylcholine is released, it binds to its receptor on the postsynaptic membrane, or in the case of muscle, on the muscle cell. So what we've learned in the neural physiology part still applies to the neuromuscular junction. So here's a blow up of this neuromuscular junction. We have a somatic motor neuron right here, and that is the axon terminal. You can recognize the vesicles that contain acetylcholine, and acetylcholine is released in response to the action potential, the influx of calcium, and then the exocytosis of the vesicle's content. Now, I want to bring your attention to this motor end plate. So see here, that's the motor end plate. So the motor end plate is a part of the sarcolemma right here, where the motor neuron meets the muscle cell. The key characteristic here is that the channels that you see here are not voltage-gated sodium channels. These are nicotinic channels, very similar to the ones that we saw in neural physiology. The implication here is that nicotinic channels are not able to produce an action potential. To produce an action potential, we need voltage-gated sodium channel. So at the motor end plate, because there's no voltage-gated sodium channels, we will not have an action potential. All we will have is this end plate potential, that first depolarization, that first graded depolarization. And then that's the end plate potential, or EPP, that leads to the action potential. So let's recap here. The somatic motor neuron releases acetylcholine inside that synapse, inside the neuromuscular junction. Acetylcholine binds to the nicotinic channels in the motor end plate. And these channels are actually cation channels. So that means that they're not specific to sodium. Both sodium and potassium can move. So when these channels open, sodium comes in based on its electrochemical gradient, but potassium will come out also based on its electrochemical gradient. Now, the reason why this produces a depolarization is that there's way more sodium that comes in compared to potassium that comes out. So we have a lot of sodium coming in, so positive ions coming in, and only relatively few potassium ions coming out. So the end result is still a cell depolarization, which, as we talked about, is called the end plate potential. Now, this end plate potential will depolarize the cell enough to reach threshold, and that will start the action potential on the rest of the sarcolemma. So it's not shown here, but the rest of the plasma membrane of the muscle here does express voltage-gated sodium channel, therefore action potentials are possible in the rest of the muscle cell. So this is a blow-up of the nicotinic channels that we just talked about. Remember that they are responsible for these end plate potentials, that first depolarization in skeletal muscle. So what we are looking at here, at the top, we're into the extracellular fluid. So that's where the synapse would happen. So that's the neuromuscular junction. This is the sarcolemma of the skeletal muscle. So specifically here, we're at the motor end plate. And then at the bottom in that yellow, we're inside the cell or in the sarcoplasm. So when our muscle cell is not stimulated, this nicotinic channel is closed and neither sodium nor potassium can move. When acetylcholine binds to this channel, it opens. And as we know, sodium comes in based on its electrochemical gradient and potassium comes out based on its electrochemical gradient. The one thing missing from this cartoon is the relative extent of that movement. And as mentioned in the previous slide, there's way more sodium that comes in compared to potassium coming out. So that's what I'm going to make a big arrow for sodium, and I'm going to leave a relatively small arrow for potassium. And that's why the end result is a depolarization, which we call the end plate potential. Now, just like any other synapse, it's good to turn the signal on, but we also need a mechanism to turn the signal off. And in this case, it's a mechanism that we're already familiar with, and it's acetylcholinesterase. Remember that acetylcholinesterase is an enzyme that breaks down acetylcholine into acetyl and choline, so two inactive metabolites. And the action of this enzyme is really important because it prevents uncontrolled muscle contraction. You know, we all had a muscle cramp at some point, maybe in our leg, while doing sport, where you feel that painful spasm and uncontrolled contraction. Imagine if this happened with your respiratory muscles. That would actually prevent you from breathing. And there are some toxins that actually inactivate acetylcholinesterase. And what that does is that acetylcholine is not destroyed at the neuromuscular junction, and you have literally muscle paralysis. If this happens with your respiratory muscle, this can actually lead to respiratory failure and death. So now we've talked about step 1, we are moving on to step 2, which is called excitation, contraction, coupling. So let's pause here and think about what this means. Excitation refers to the action potential, and contraction refers to muscle contraction. Coupling means link. So here we're trying to explain how do we get from an electrical signal or an action potential signal and translate that into an action, which is the contraction. To do that, the cell will have to translate the action potentials into a calcium signal. So let's see how that's done. To understand this, we need to recall that there is a physical interaction between the T-tubules and the sarcoplasmic reticulum. And that's what we see in the cartoon here. So in yellow, we have the sarcolemma of the muscle cell. So that's the plasma membrane. But as you can see, instead of being continuous here, there's a big dip, which we call the T-tubules. We see 2 T-tubules in this specific cartoon. In blue, we have the sarcoplasmic reticulum. And you can see that there is physical contact between the T-tubules and the sarcoplasmic reticulum. Now, the other thing that we should remember is that the sarcoplasmic reticulum's main function is calcium storage. So inside that blue structure, inside the sarcoplasmic reticulum, there is a very, very, very high concentration of calcium. So the physical link between the T-tubules and the sarcoplasmic reticulum is made via 2 proteins. The first one is called DHPR, the second one is called RYR. So I'll introduce them here and we'll see them in the next couple slides. The DHPR is part of the sarcolemma. So it's on the T-tubule. So DHPR is on the T-tubule. The other protein is the RYR, which I'm going to draw here in red, and that is on the sarcoplasmic reticulum. So when we talk about physical contact between the T-tubules and the sarcoplasmic reticulum, this happens because these two proteins touch and interact one another. So that the DHPR on the T-tubule interacts with the RYR on the sarcoplasmic reticulum. So let's blow this up and see how this works. So we have our T-tubule right here, and we have our sarcoplasmic reticulum here in purple. And the two proteins that we just introduced are here too. So the DHPR is here in dark blue, and the RYR is here in light blue. And you can see with this squiggly black line that there is a physical contact between the two. So let's start with the DHPR. The actual name is dihydropyridine receptor, or DHPR. The DHPR protein is sensitive to voltage, but it's not a protein that opens and closes like a lot of the channels that we've seen. What it does in response to depolarization, it changes shape. The fancy word for that is a conformational change. So as the action potential comes down the T-tubule, the DHPR changes its shape in response to voltage. This conformational change causes the RYR channel to open. As an analogy, think about it as if the DHPR was pulling on the door of the RYR. So the conformational change of the DHPR happens first, which then causes the opening of the RYR. The RYR are calcium channels. So all the calcium that's stored inside the sarcoplasmic reticulum in really high concentration is now allowed to flow out down its electrochemical gradient. So what happens is that calcium inside the sarcoplasm goes way, way up. Calcium inside the sarcoplasm can actually go up 100-fold during muscle contraction. So this slide really explains how we get from an electrical signal, which is your action potential, to a calcium signal. And remember that this process is called excitation, contraction, coupling, or the link between the electrical signal and the calcium signal. So just to reiterate here, in a muscle at rest that is not contracting, most of the RYR proteins are closed, which means that calcium stays inside the sarcoplasmic reticulum and the concentration of calcium in the intracellular fluid in the sarcoplasm is low. Contrast this with a stimulated muscle. In this case, RYR is open and calcium flows out of the sarcoplasmic reticulum. Therefore, the concentration of calcium inside the sarcoplasm is high. So in our next video, we'll discuss how calcium allows the interaction between actin and myosin and the contractions of muscle. Stay tuned.